let us move on to the next speaker. Who is it one? No, that is over. Professor, I think first one we'll just talk about. No? Professor Ikayare you ready?
Yes, I'm ready.
Okay, please talk.
Okay, thank you. So I think this time it shouldn't be any problem, so you can see it clearly?
Yes.
Yes.
Thank you. So I will quickly start my talk as I've just introduced myself. I am from Faculty of Pharmacy, Gazi University in Ankara, and I will focus on neurobiological activity of natural products, also based on our experience. As you know, plants are very important biological sources in drug discovery, not only drug discovery, but also cosmetics industry. And according to NAPRELAR database, actually only 15 to 20% of all plant species were investigated so far for their phytochemistry, whereas only 5 to 10% of these plants have been screened for their bioactivities. So we have still a long way to go. On the other hand, number of the molecules, promising drug candidates or clinically available drugs entering in the drug market is decreasing because it's very costly and time-consuming procedure. So in that sense, my main focus is with plant phenolics and their neuroprotective activity. And when we just, popped in PubMed like that, we can already find like almost 15,000 results. There are a lot of research going on in this area. And if we limit it to plant extracts and neuroprotection, we still get like 5,000 paper on that. So it's a very popular subject in the scope of pharmacognosy, as I can say. So as I've just mentioned, so our focus is Alzheimer's disease, and of course, it's a very complicated neurological disease, which is explained by two theories called amyloid plaque theory and cholinergic hypothesis. So besides these two main hypotheses to explain pathogenesis of the disease, there are some other mechanisms, of course, pathophysiological mechanisms, including oxidative stress or hyperphosphorylation of tau protein, as well as accumulation of metal ions. These are some other side pathologies associated with this disease. So, of course, there are lots of enzyme playing role in the pathology of this disease. And in that way, related to amyloid hypothesis, actually, there is one enzyme family very critical, which is secretase family. And among them, beta secretase is quite important because it has function to cleavage the protein called APP or amyloid protein, I can say. So anyway, inhibition of these secretase, beta secretase, is another treatment strategy for Alzheimer's disease, because they cause, if they are not inhibited, they cause amyloid beta protein to accumulate in the brains of Alzheimer's disease. So what is workflow in our laboratory? Actually, we still rely on folk medicine or traditional medicine. Sometimes, we collect the plants according to their folk medicinal use, but sometimes we also do random screening. Anyway, we collect the plant, we do extraction, and through the extracts, in each step, we do some enzyme inhibition assays. So when we get active extracts, then we apply the method called activity-guided fractionation, which leads finally to isolation of active compounds. But it's at each step, we apply these enzyme inhibition assays. So our aim, as you know, to find an active compound or compounds. So after doing phytochemical analysis, we basically proceed them to molecular docking experiments and sometimes in vivo experiments, and sometimes some cell culture experiments. So for determining neuroprotective or neurobiological activity of plant phenolics or natural products in general, so we we applied methods, in vitro methods, using some enzyme inhibition assays, and we have in vivo methods like passive avoidance test in mice, which is available in our lab. We also collaborate to do experiments on zebrafish and Drosophila melanogaster brain homogenate. And as I said, we use in silico tools to see the interactions between the active compounds and the interaction sites of these enzymes. And if available, of course, we proceed them to cell culture assays as well. So the first work that I would like to mention is on Cassia tora. So this is an Indian plant, which is locally known as Takrika. So this was a joint work with Jain University in India. So the fresh leaves of this plant were collected, and we prepared several extracts in different polarities, and we just subjected these extracts to anti-amyloidogenic activity to see how they affect this amyloid beta 42 peptide through inhibition of oligomerization, fibrillation, and defibrillation of this protein using thioflavin T fluorescence assay, and also monitoring the results by transmission electron microscopy. Of course, also doing tests on their cholinesterase inhibitory activity if possible. So for this assay, actually, when we tested the extracts at the first place, here you can see the decreased fluorescence intensity means the reduction in beta structures of aggregates located in these plaques in the brain, in fact. So these results show that actually the petroleum ether and aqueous extracts of this plant had some activity. Then the hexane extracts was not really active because it didn't lineate the intensity of fluorescence. And the most active one was actually CT methanol extracts, which inhibited amyloid beta protein fibril formation. Then we monitored these changes through team analysis, and here you can see in versus control group, actually, these methanol extracts show clearly the absence of, amyloid beta aggregation. And this factual decrease can be seen here again in the end of this arrow, in formation of A beta protein. So after 72 hours, we also observed the effectiveness of CT methanol extracts to see if there is an inhibition on formation of these beta fibril-like structures. And the extract again caused a substantial decrease in intensity of fluorescence versus control. So we again came to conclusion that this extract was active, and again, doing team analysis with the extracts at different time periods. Here you can see after 24 hours, actually the extract delayed formation of these oligomers here. And after 72 hours, we can say that the oligomers were almost absent. And after 120 hours, there was almost no fibril formation caused by Cassia tora methanol extracts. And then in the further analysis using THT assays, after incubation of these extracts with the peptide during 9 days, again, we can clearly see the effective inhibition of this formation of fibrillar structure, major fibrillar structure of synthetic this beta peptide. Then here again in team analysis, we can see in defibrillation part of this assay, actually, the CT methanol extracts was moderately active, I can say. But anyway, after evaluating all these cumulative data, we can say that this methanol extracts obtained from Cassia tora was able to inhibit aggregation of this beta peptide from oligomers, and also it can disaggregate this fibril formation. After that, we tested all these 4 extracts in cholinesterase inhibitory assays, and here you can see the methanol extract was again consistently effective to inhibit this enzyme, causing inhibition over 50%. Then we further analyze these extracts using HPLC, and this is fingerprint analysis showing the phenolic compounds. And here you can see well-known phenolics like flavonoid, rutin, and quercetin, and caffeic acid, paraquinaric acid, chlorogenic acid were the main ones. And when we determined the phenolic and flavonoid content, total contents of these extracts, again, methanol extract was had the most abundant amount of these compounds. So in another project of our group, we also screened many plant phenolics, almost 36 or 37, as you see here. We firstly, in the first step, we tested them against cholinesterase enzymes. These are sister enzymes playing role in cholinergic hypothesis, and here you can see some of them were quite promising, like gallic acid, caffeic acid, and rosmarinic acid, as expected. And hydroxytyrosol was not tested before anyone else, so we also showed that it is quite effective, and trans-resveratrol, quercetin, EGCG. And in the second part, you can see the results for, promising results for fisetin, apigenin, other flavonoid derivatives, and taxifoline. So they were quite promising. Since accumulation of metals in the brains of Alzheimer's patients is another factor to trigger the disease and also causing neuroinflammation. So we tested these compounds for their metal chelation capacity. And as you can see here, versus ETA used as a reference, the compounds, these phenolics were not that active. Actually, they caused only active, metal chelation capacity below 50%. The most active one was tangeratin. And then we also tested their antioxidant activity using FRAP and QPRAP assays. As you can see, we used quercetin here again as a reference, and some of these compounds were quite active in FRAP and QPRAP assays, having antioxidant value identical or even superior to that of the reference. Then we pushed these compounds, some of these selected compounds, to in vivo experiments. So what was that? Was anti-amnesic activity induced by scopolamine in mice. So for this purpose, we use an apparatus called step-through type device, which is available in our lab. So we basically, as you can see here, mice always tend to go to dark places, dark holes. So this device has two compartments. One is dark and another one is with light. So of course, when we put the mice in the device, so they tend to go to dark place. But when they go to dark compartment, we apply some little electroshock. So they feel some dizzying in their feet. So they actually, we just try to teach them that they should fear, because there is danger in dark compartments. So basically, after applying our samples, test samples, we just measure the latency time. So the latency time should be longer if they think that there is danger over there. So this is basically the logic behind this experiment. So in this anti-amnesic activity experiment in mice, here you can see the latency time for the first day and second day. So second day latency time for all these tested compounds were longer, much longer, and statistically meaningful because these 5 plant phenolics were quite active. And here in this graph, you can see the difference on latency times caused by test samples. So they definitely seem to be very active. And then we push the selected compounds to another in vivo model using Drosophila melanogaster. So because this is very useful organisms and also used in biological research since last century because it has 70% of homology to human genome. That's why it gives some meaningful results for human diseases as well. So using this fruit fly, we tested the compounds on your left. We fed Drosophila larvae with this phenolics at 2 concentrations. So for this purpose, we spent like 9,000 adult individuals of Drosophila, and then we removed their heads and homogenates it, and then we just measured the cholinesterase activity in their brain, and here you can see the Vmax plots of these plant phenolics for their cholinesterase, acetylcholinesterase activity in comparison to another drug, galantamine, as a reference drug. This is the Vmax plots, and we just saw after this experiment that 6 out of these 9 studied plant phenolics, 6 of them were quite active. So these were rosmarinic acid, resveratrol, gallic acid, oleuropein, 3-hydroxytyrosol, and toxipolin. They were the most active ones. So then we did some analysis called GWAS, genome-wide association studies, with these compounds. So through this GWAS analysis, it is possible to see the candidate genes or pathways linked to Alzheimer's disease or cholinesterase inhibition. So anyway, here you can see the responses. I'm not going to go into details, but from these results, we can again see that Vmax values for these compounds were much lower for butyrcholinesterase than for acetylcholinesterase, which was consistent with our data obtained in vitro microplate assay for these enzymes. So through GWAS analysis, we could see the results, like these are the candidate genes with which these phenolic compounds can interact. Anyway, I'm just going too quick, I'm so sorry. So with these 8 compounds, 8 effective compounds, we just wanted to see how the gene levels are influenced, especially the 14 genes related to Alzheimer's disease. So among them, I can say that quercetin, oleuropein, EGCG, which is a very well-known green tea polyphenol, as you know, and hydroxytyrosol, they showed or exhibited similar patterns in the change of gene levels of 13 genes, as you can see here. And in second party, these 4 more polyphenolics were also tested in PCR to see gene expression level, how they affect. So from this result, I can say that fisetin can decrease the expression of APP and PSEN genes, which are the most studied genes for Alzheimer's disease. Just going through, and here you can see the cumulative data for this gene expression experiment, and fisetin seems to be very active here. Then we tested their
2 minutes, please.
Okay, I'm just going to the end. So cell viability results, we can see they were quite cytotoxic in neuroblastoma cell, and here you can see the IC8 values for these compounds at micromolar level. Then we pushed them into molecular docking experiment. I'm not giving the detail, but here you can see that this just an examples. Of course, we did all these experiments for all these active compounds, but some experiments for some of these compounds like rosmarinic acid, hydroxytyrosol, and gallic acid, they show different, not very different, but similar actually interactions with the amino acid residues, like some of these 2 amino acid residues, G120 and Y133, for acetylcholinesterase active gorge. And for butyrcholinesterase, again, for the same 3 compounds, they exhibited different interactions through, hydrogen bonding or etc. Here you can see the interactions with some of these amino acid residues. So we did in silico estimate of potential biological activity of these compounds. Just an example, rosmarinic acid, gallic acid, and hydroxytyrosol showed very good probability for amyloid beta aggregation inhibition using Swiss target test program. Then in silico toxic and adverse effects, which might be expected in humans for these compounds using Vega, QSAR, and TOX3 models, and they show this may show in silico environment as toxic and adverse experiments. So this is the end of my talk. This is my lab team and collaborators. I would like to thank all of them, and most of these studies actually were supported by our university scientific research project unit. I am also grateful to Turkish Academy of Science giving me the budget to do this research. And thank you very much for participating in my talk. And if you happen to come to our beautiful capital, Ankara, so we will be very happy to host you. Thank you very much again.
Thank you very much. Thank you very much for that stimulating presentation.
Thank you, Professor. Your talk is very, very excellent. And nowadays, this Alzheimer is increasing day by day. And you have really tried to extract the plant active pigments or the plant extract, polyphenols, and then you are really trying to, because there are so many medicinal plants which can be used to prevent such type of the diseases. And not only the medicinal plant, underutilized plants also can be make use of it for the such type of the medicinal. Thank you very much.
Thank you very much. This was very quick, but anyway, thank you.